So hello everyone, we are here with a new topic of microbiology, that too from the topic of the parasitology, that is the Entamoeba histolytica. So first we will complete one or two parasite, the Entamoeba histolytica and some other parasite, and in a separate video I will be teaching you the general parasitology. So now moving to the Entamoeba histolytica, first of all we will see the morphology. So in the morphology condition, what happens now, in the Entamoeba histolytica, it exists or it shows its morphology with the help of the three stages. There the first stage is called the trophozoite, or it is also called the vegetative form, or also it is the feeding stage of the parasite. Second is the pre-cystic stage, and the third is the cystic stage. So the pre-cystic stage is nothing but it is the intermediate stage or the transforming stage from trophozoite to the cystic stage. Okay, done. Then moving to first the trophozoite stage. So in the trophozoite stage, what happens? First of all, the trophozoite stage, the morphology, we will see the morphology in the separately in the trophozoite stage, in the pre-cystic and in the cystic stage. So first in the trophozoite stage, the morphology is, it is irregular. The size varies from 10 to 40 micron. Average is the average size is 20 micron. Okay, and then it is then talking about the motility of the parasite in the trophozoite stage. It is actively motile with the help of pseudopodium. It is actively motile with the help of pseudopodium. Then moving to the cytoplasm of the parasite in the trophozoite stage, it exists in the two forms, the ectoplasm and the endoplasm. In the ectoplasm it is thin and clear, but in the endoplasm it is granular, which contains nucleus, the food vacuoles and the RBCs, and sometimes in some cases it also contains the white blood cells. Then after cytoplasm, moving to the nucleus of the parasite in the trophozoite stage, it is spherical, having a size of 4 to 6 micron. The appearance is, it gives a dot-like structure in the center. It contains some dot-like structure in the center, that is called the karyosome. Some dot-like structure in the center, that is called karyosome, and it is surrounded by nuclear membrane, which is lined with a single layer of uniformly distributed chromatin granules. Okay, the nucleus is spherical with a size of 4 to 6 micron, dot-like structure present in the center called as karyosome, and it is surrounded by nuclear membrane lined with a single layer of uniformly distributed chromatin granules. And moving to the next page, after the trophozoite stage, it is the pre-cystic stage. As I have already said earlier, it is the transitory stage from trophozoite to the cystic stage. Okay, and the morphology in this stage is, it is round or oval with blunt pseudopodium. Round or oval with blunt pseudopodium, size from 10 to 20 micron. It is only having the endoplasm, which is free of RBC and the other ingested particles. In the previous page we have studied here that the endoplasm contains RBC, WBC, but here it is free of RBC and other ingested particles. And then moving to the nucleus of the parasite in the pre-cystic stage, there is no much change. It retains all the features of the trophozoite stage. Okay, now moving to the cystic stage. The cystic stage, it is, first of all, it is spherical. The size is 10 to 15 micron. Here the cytoplasm is nothing but a mass of glycogen. Here the cytoplasm is a mass of glycogen, and 1 to 4 cigar-shaped chromatoid bodies are present in the immature cyst. In the immature cyst, first there are, it's a mass of glycogen, and 1 to 4 sugar-shaped chromatoid bodies are present. But as soon as it gets mature further, the glycogen mass and the chromatoid body starts to disappear. Then moving to the nucleus, each cyst contains a single nucleus. Okay, and after then it undergoes the 2 successive mitotic division to form 2 nucleus, finally 4 in the mature cyst. So the mature cyst is a quadrinucleated state. Done. So it is the stages. Look here. First of all, it is the first stage, the trophozoite stage. Here it contains the ingested food particles, the pseudopodium. Here is the nucleus, and all set. Next, it now moves to the pre-cystic stage, where all such ingested particles are absent. No ingested particles, only nucleus is present. Then it undergoes further division or differentiation. Then it forms the uninucleate cyst, or it is the immature cyst, which contains the chromatid bars. After that, it undergoes the first mitotic division to form binucleate cyst, and then the second mitotic division to form the quadrinucleate cyst, and it is the matured cyst. Okay, it is the matured cyst. Done. Okay, now moving to the life cycle. So now moving to the life cycle. So in the life cycle of the Entamoeba histolytica parasite, here we have the definitive host, that is the human being, but there is no requirement of the intermediate host, as it occurs directly between the host and the parasite. Matlab the definitive host and the parasite direct it occurs. Okay, acha. So it is the life cycle of Entamoeba histolytica. So let's see it. First of all, what happens now? The mature cyst. Okay, the mature cyst passes in the phases of convalescence and the carrier contaminated food and drinks. If anyone is a carrier of this parasite, and the food and drinks get contaminated with that carrier, so in that carrier contaminated food and drinks and the feces of that infected persons, that is the mode of transmission. So now it enters into the human body. The infection occurs by ingestion of contaminated food and the drinks. Now what happens now? Once it enters the human body, the cyst passes through the stomach undamaged and enter the small intestine. Stomach, then it enters now the small intestine. Now what happens now? In the cecum or in the lower part of the ileum, or in the cecum, excistation occurs. So what happens during the excistation? And during excistation, the cytoplasm gets detached from the cyst wall. The cytoplasm gets detached from the cyst wall. Then now it causes something to the cyst wall, which is a tetranucleate amoeba comes out from. Then the cyst wall got ruptured and a tetranucleate amoeba comes out, which is the metacyst. Now after the metacyst got released outside from the cyst wall or from the cytoplasm, whatever may be, then now the nuclei and the cytoplasm of the metacyst, it's a tetranucleate amoeba. So now which is a metacyst? Now ये जो मेटासिस्ट है, इसी मेटासिस्ट का न्यूक्लियस और साइटोप्लाज्म अब डिवाइड होके बनाएंगे क्या? 8 amoebulae which lodge in the submucous tissue of the large intestine, where they grow and multiply by binary fission. See, large intestine के submucous tissue में अब वो रहेंगे, ग्रो करेंगे और मल्टीप्लाई करेंगे with the help of binary fission. after also such sorts of things, it produce the characteristic lesion of amoebiasis. Now produce the characteristic lesion of amoebiasis. When conditions become unsuitable, for the survival of the trophozoite form, if such a condition arises that is not suitable for the survival of the trophozoite form, then they gets converted to the pre-cystic stage. Then finally into the cystic stage. Okay, pre-cystic stage and finally into the cystic stage. And the cystic stage are passed in the phases again to repeat the cycle. That means the infected stage is the trophozoite stage. When condition is unfavorable, the trophozoite form gets converted to pre-cystic, then into the mature cystic stage. Then it again passes through the phases and repeat the cycle. This is the life cycle of Entamoeba histolytica. So now moving to the pathogenicity. What happens now in the pathogenicity? Unless until the trophozoite is in the lumen, no illness occurs, no such disease condition arises. But when the trophozoite form, which is the infectious form or the infective stage of Entamoeba histolytica, when it invade the intestinal tissue, there occurs the disease. And it happens only in the 10% cases, because 90% of cases remains asymptomatic. Okay, 90% of cases remains asymptomatic. Next, let's see the pathway of the disease development in infection by Entamoeba histolytica. So what is the process of disease development now? When the metacystic trophozoites invade the mucous membrane of large intestine. When the metacystic trophozoites invade the mucous membrane of the large intestine, it causes damage to the mucosal epithelium with the help of the motility and the proteolytic ferment secreted. Okay, when the metacystic trophozoites invade the mucous membrane of the large intestine, it cause damage to the mucosal epithelium of the large intestine with the help of the motility and the proteolytic ferment secreted by the trophozoite stage. Now, as soon as it reaches the submucous coat, it gets multiplies and increase in number and form the colonies. It multiplies, increase in number and form the colonies. After formation of the colonies, in increasing in the number and multiplication, they destroys the tissue in their vicinity and utilize the cytolized material as their food. They destroy the tissue or the cells in their vicinity, within their area or within their capacity, and after the destruction, the cytolized materials become the food for the parasites. And after getting the food, now they are energized. They are energized. They got the food, so now they spread rapidly in various direction, and now they dissolve the surrounding tissue and bring about the coagulative necrosis. First, within their capacity, whatever tissues or cells are there, they cause the destruction of that tissues, and with that cytolized materials, they consume them as their food, and now they become powerful, they become energized and spread rapidly in various direction and dissolve the surrounding tissues, also bringing coagulative necrosis. So due to coagulative necrosis, there is the formation of abscess, which breaks down to form an ulcer. So what happens in the ulcer? So what type of ulcer? What is the size, the shape, where is the margin, how is the base, how it looks like? Let's see the ulcer which caused due to Entamoeba histolytica infection. The ulcer is multiple and confined to the colon only. It confined to the colon only, and it's not single ulcer. There will be multiple ulcers. The size is from the head of the pin head to equals or more than 1 inch. From pin head to more than or equals to 1 inch. The shape is round or oval, but in the cross section it appears flask shaped. Generally it is rounded oval overally, but in the cross section we will see the flask shaped appearance. What is the margin? The margin is ragged or undermined as like in the tuberculosis ulcer. The shape will be like this. Matlab from outside it appears to be very small, but inside it's very deep. That is the undermined or ragged ulcer. And the base is generally formed by the muscular coat and filled up by the necrotized material. The base is formed by the muscular coat and filled up with the necrotic material. So after the ulcer, it also spreads to the different organs. So now after the formation of the ulcer, now it will spreads to the other organ. So what is the pathway of spread? First of all, there will be the invasion of the intestinal wall by the amoeba. Next, now the amoeba, after the invasion of the intestinal wall, it gets penetrated the radicals of the portal veins. Portal veins के radical branches को ये penetrate करेगा, and after the penetration it gets transported through the blood to the liver. पोर्टल वेन पार्टिकल्स की वजह से अब वो पेनिट्रेट करने के बाद अब ये ब्लड के ज़रिए होके लीवर तक पहुंचेगा. आफ्टर गोइंग रीचिंग द लीवर, it gets established in the hepatic lobules where they will multiply, and after the multiplication, now there will be initiation of the lytic necrosis. There will be initiation of the lytic necrosis with inflammatory reaction. Obviously there will be inflammatory reaction which causes the obstruction to the circulation and produce the thrombosis of the portal venules. After establishment in the hepatic lobules of the liver, where the parasite or the amoeba will multiply, it will initiate the lytic necrosis with the help along with the inflammatory reactions, which will cause obstruction to the circulation and produce thrombosis of the portal venules. As a result, there will be anemic necrosis of the surrounding liver cells, which is called as miliary abscess. Yes, the anemic necrosis of the surrounding liver cells due to obstruction of the circulation and thrombosis of the portal venules is called as miliary abscess or miliary abscess. Sorry, it's not miliary abscess, it's miliary abscess. And now what happens now, when more than one miliary abscess formed, the miliary abscess coalesce with each other. Coalesce means they combine or join to form an abscess which varies from size from few millimeters to few centimeter, containing a very thick chocolate brown pus which is called as anchovy sauce pus. Anchovy sauce pus, which contains, the content is liquefied necrotic liver tissue and blood. Okay, it's important, you have to remember this. What is the miliary abscess, anchovy sauce pus, the content of anchovy sauce pus. Why it is chocolate brown color? Because due to the presence of liquefied necrotic tissue, liver tissue and the blood. So after this, या तो there will be rupture of the abscess or there will be hematogenous spread. If there will be rupture of the abscess, what happens now? If the abscess will be untreated, then now the untreated abscess may rupture into surrounding organ and the tissues. If the abscess remains untreated, it may rupture into the surrounding organs and the tissues, like to the lungs, to the pleural cavity, pericardium, peritoneal cavity, stomach, intestine, inferior vena cava, abdominal wall and skin. Or it may spread to the lungs and skin by hematogenous route via blood, and the amoeba may spread to the organs. After spreading to the lungs and skin via blood, it may spread to the lung, skin, brain, kidney, spleen, adrenal glands, these all. So it is the pathway. Okay, so now what happens now? Let's move to the lab diagnosis of Entamoeba histolytica. So the lab diagnosis of Entamoeba histolytica, first step is the diagnosis of the what? First step is, so now the lab diagnosis of the Entamoeba histolytica. First, the diagnosis of intestinal amoebiasis or the amoebic dysentery. Here what we will do now, the amoebic dysentery has to be differentiated from the bacillary dysentery by inspecting stool sample microscopically and macroscopically. After completion of this lab diagnosis entire, we will move to the differentiating point. First of all, there will be diagnosis of hepatic amoebiasis. Hepatic amoebiasis, what we'll see now, for this the sample will be the pus aspirated, yes, the pus aspirated from the liver or the liver biopsy is observed for the presence of the trophozoites, whether trophozoites are presence or absence, that we will observe from the pus which is aspirated, matlab injected out with the help of injection or syringe from the liver or liver biopsy. Next, the stool sample may shows the presence of the cyst in about 15% of cases. Next, we have to do the serological test. The positive test indicates the presence of specific antibodies in the blood, and the antibodies can be detected by the complement fixation test, CFT, agar gel diffusion, latex agglutination, indirect hemagglutination test, then the ELISA, immobilization test, and indirect immunofluorescence test. And for the detection of the antigen, the antigens can be detected by RIA, the radioimmunoassay. And the amoebic antigens in the serum are also detected by using ELISA, the coagglutination test. So here is a type of the test written CIEP. Here is also written CIEP. The full form is the counter current immunoelectrophoresis. It is a rapid laboratory technique used to detect specific antigens and or antibodies in the clinical samples such as blood or CSF. So this is all about the diagnosis of the hepatic amoebiasis. Next is for the steps. So what are the steps for the diagnosis of the intestinal amoebiasis? So first we have started the diagnosis of intestinal amoebiasis or the amoebic dysentery. So for this we will see the differentiating feature, but let's say the collection and the transportation of the specimen. First of all, what are the specimen? And the feces. The fecal matter, the feces are collected and transported immediately, and then the next step is for the microscopy. In the microscopy, there is the saline preparation and the iodine preparation. Saline preparation, that is to demonstrate the mobile or the motile, the trophozoite, and the iodine preparation is done to demonstrate the trophozoite in killed state and the cyst in the staining state. Next, the detection of amoebic antigen. It is done by using that same, the CIEP, the counter current immunoelectrophoresis test, and by ELISA using monoclonal antibodies. Last is the serological test. It is negative for antibody in the early cases and in the absence of deep disease. The second was our diagnosis of the hepatic amoebiasis, which we have completed. The third is the diagnosis of pulmonary amoebiasis. In pulmonary amoebiasis, we have to see the demonstration of the trophozoite in Encovis sputum. लीवर और लंग से ही हमारा आता था ना एंकोविस स्पुटम, तो एंकोविस स्पुटम. The Entamoeba gingivalis normally present in the mouth may cause confusion in the diagnosis. They may cause confusion in the diagnosis, so there will be diagnosis by the serological test. And what further, the newer technique in the diagnosis of the amoebiasis, the molecular method. Detection of Entamoeba histolytica DNA in the stool by using DNA hybridization probe, and also the PCR, polymerization chain reaction, is done to detect the genome in the pus or the stool. Now moving to the differentiation between what? The differentiating features between amoebic dysentery and bacillar dysentery. What happens now? First we will see the macroscopic features, then the microscopic features. In the macroscopic features, first the number of motion or the loose motion in amoebic dysentery, it is 6 to 8 per day. In bacillary dysentery, it is more than 10 per day. Odor of stool is offensive in amoebic dysentery, it is odorless in bacillary dysentery. Color of stool in amoebic dysentery, it is dark red. In bacillary dysentery, it is bright red color. The nature of the stool in amoebic dysentery, it is mixed with blood and mucus, but in bacillary dysentery, just blood and mucus will come out, no fecal matter will come out. The main differentiating point. Consistency. In amoebic dysentery, the fecal matter doesn't remain adhered to the container, but in bacillary dysentery, it is adherent to the container. And the amoebic dysentery, it is acidic and it is alkaline in bacillary dysentery. Moving to the microscopic features, there is reddish yellow color in the clumps, the presence of RBC in clumps, but in bacillary dysentery, the bright red color due to the raw legs formation. In amoebic dysentery, the pus cells are present in very few number, very scanty, but it is in a large number, numerous in bacillary dysentery. Macrophages, a very few number of macrophages can be found in amoebic dysentery, but several macrophages can be found in bacillary dysentery with ingested RBC. Then the eosinophil, the Charcot-Leyden crystal, motile bacteria, amebia, they are all present in amoebic dysentery but absent in bacillary dysentery. Moving to the final, what is the treatment for Entamoeba histolytica infection? The drugs are metronidazole, diloxanide, furet, then the diiodohydroxyquine or the paromomycin, that is generally for the trophozoite as well as the cyst. Then in amoebic colitis and amoebic liver abscess, metronidazole, tinidazole, secnidazole, ornidazole are effective. Chloroquine, tinidazole and the tetracycline are all affected in the hepatic and pulmonary amoebiasis. It should be remembered. And this is all about our Entamoeba histolytica, and this completes the chapter. So we will meet again with a new chapter with a new topic.